I'm Xiang from Yeasen and Molefuture. And I'm very glad today to give a report about our latest research on T7 RNA polymerase engineering. So today, my talk will be two parts. Firstly, I will introduce a bit about Yeasen and Molefuture, which is a subsidiary of Yeasen. And then we will come to our main topic today about the engineering of T7 RNA polymerase. We reduce dsRNA content. So about Yeasen. Yeasen is a Chinese company based in Shanghai. We focused mainly on developing raw materials, mainly about enzymes and also antibodies and related life science bioreagents. Based on our technology platforms and production facilities, we also developed multiple product lines in the industry of life science research, in vitro diagnosis, and biopharmaceuticals. Our main cutting-edge technology platform include enzyme-directed evolution and ultra-clean enzyme production, tailor-made for molecular enzymes. We will mainly talk about this technology platform today. So our headquarters is located in Shanghai, and we have production facilities in Wuhan, which is in the middle of China. And then we have also other subsidiaries in other countries, like in the US, in Maryland, and also in Singapore. After 10 years accumulation of technologies, we have developed 6 core technology platforms in enzyme development areas, starting from the enzyme editor engineering, enzyme engineering platform, which mainly drives the enzyme innovation. And we also have multi-host expression platform, fermentation platform, purification platform, the ultra-clean production platform for GMP-like level enzyme production, and also the analysis platform for all kinds of different molecular enzymes. So one of the significant technology platform is our enzyme editor enzyme engineering platform. We all know that enzyme is a protein, which is the sequence of which is very important for the function. So our platform applies two important technical routes to involve enzymes. One is the directed evolution, and the other one is the rational design. By combining both routes, we can engineer an enzyme with desired properties in a short time and a higher success rate. Because enzyme is also a fundamental element of the biotech technology area, so this platform mainly can not only can work on molecular enzymes, it can also work on different enzymes. In recent years, we have developed 2 platforms for enzyme engineering. One is based on the high-throughput screening method establishment, and the other one is for the rational design. So for the directed evolution technologies, we mainly we have mainly 3 technologies. One is the fluorescence activated droplet sorting, which is ultra high throughput screening using fluorescence and microfluidic technologies. And we also have the traditional microtete plate screening. As we all know, for direct evolution of industrial enzymes, they don't need to purify the enzymes. We can use the lysate to do the analysis, to do the screening. But for molecular enzymes and also pharmaceutical enzymes, we need to purify the enzyme before the assay. So we developed a high throughput purification platform to incorporate into this traditional microtubule screening method, so that we can work on most molecular enzyme to do the 96-well plate screening to make it feasible. The last high throughput screening technology is the compartmentalized self-replication, which is tailor-made for DNA replication-related enzymes such as DNA polymerase. This is also special for Yeasen because most of our enzymes can be applied in this technology platform. So we all know that rational design or machine learning is very popular in the worldwide. So we also have this computer-aided rational design, which applies the structure and structure function relationship, as well as the cutting-edge machine learning and artificial intelligence technologies, because we have generated a huge of wet data from our direct evolution platform, so that we can learn from it and do the better prediction of new functions or improved functions. So based on our technology platform, especially the enzyme editor, enzyme engineering platform, so we founded Molefuture last year as a subsidiary of Yeasen and focusing only on the enzyme tailor-made enzyme solutions for different industries. So our core business includes mainly the enzyme engineering, using our technologies to improve the activity or selectivity or stability of enzyme from different industries. This happens when you have an enzyme or you have an enzyme process to start with. If you don't have an enzyme, we can also provide the discovery of enzyme service to help you to do the enzyme mining using all kinds of the open database as well as AI tools. After we have a molecule with sufficient properties, we can also provide the process development. For example, the fermentation process optimization and also the purification process optimization. At last, we also can do the scaling up of molecular enzymes, such as for the pharmaceutical enzymes, because we have a GMP-like production site in Wuhan, which meets the request of the biopharmaceutical needs. So as I just mentioned, enzyme is the very fundamental element of biotech. So basically, our enzyme editor, enzyme engineering platform can support all kinds of different industries. We know that synthetic biology is very popular now worldwide. We can provide enzyme engineering service to those who want to engineer their enzymes in their pathways. Also for the synthetic enzymatic DNA synthesis, also for different different purposes. We can also engineer enzymes for pharmaceuticals, biopharmaceuticals, also for the API, and also for farm chemicals, health and nutrition, industrial or specialty enzymes. So basically, all industries who want to engineer enzymes, we can do for them. The properties to be involved not only about activity, stability, but also about affinity, the pH or organic solvent tolerance, or different selectivities of enzymes. So in the last 10 years, we have commercialized more than 100 molecular enzymes. by Yeasen. This include in vitro diagnosis enzymes, next generation sequencing enzymes, RNA synthesis enzymes, nuclease enzymes used in biopharmaceutical processes, the PCR enzyme, PCR related enzyme in life science research, and other enzymes, which is needed for this, for different biotech industries or research. So today we will mainly focusing on the T7 RNA polymerase because we have engineering different enzymes as needed, and the T7 RNA polymerase is the most popular one now. So maybe there are a lot of background about the mRNA therapy, but today I will focus mainly on the enzyme part, especially the T7 RNA polymerase. We know that T7 RNA polymerase is very important for mRNA therapy, but it is an enzyme with different domains and different structures, different domains and different functions or different activities. The main activities help to do the transcription, but it also has side activities. For example, the promote independent transcription, terminal transferase activity, and RNA-dependent RNA polymerase. All of these side activities will lead to non-specific RNA formation. And one of the most concerned one is the dsRNA, the double-strand RNA byproduct. If it goes to the human body, it will lead to immune stress responses, which is not wanted for the therapy. So currently, there are ways of removing this using purification, but it leads to extra work, processing burden. Lately, there are also ways to prevent the formation of the dsRNA. They also some company also involved thermostable T7 RNA polymerase to prevent the formation of double-strand RNA. However, if we apply this T7 RNA polymerase, as it's we need to accelerate the temperature of the IVT process, and the other enzymes, maybe more than 10 enzymes, we also need to be semi-stable, which is actual work for a lot of evolution work. Besides, the high temperature not preferred for the process. So an enzyme with decreased dsRNA itself maybe is more favorable for the future application. So here comes the question: we want to involve T7 RNA polymerase with reduced dsRNA without changing anything else. So we need to know how the dsRNA is produced, the generation mechanisms of dsRNA, starting from here. So firstly, there are a lot of literatures talking about the mechanism of this T7 RNA polymerase. We can start from here. So basically, there is a huge conformational change during the transcription, starting from initiation conformation, and then to the elongation state, and then to the termination. So we can see from these two pictures, the first one, the initiation complex, and the second one, the elongation complex. So so after the transcription starts, like maybe less than 10 base pairs, then it will undergoes a huge conformational change. You can see the green ribbon flipped up. It's a big change, which is which most of the time maybe it will lead to failure here. So if it failed in this conformational change, it will form short RNA, which is also called abortive RNA, and then it will lead to short dsRNA. Even if the elongation is successful and it go to termination, it also will form actual long RNA products. We can see the the actual nucleotide in the 3' end, and it will form the loopback dsRNA later. So all of these two mechanisms of dsRNA formation may lead to dsRNA, but it also has some other mechanisms to form the dsRNA. For example, the complementary pairing with fermenter-independent transcripts shown here. But we cannot do all of things at once. So in our research, we focused on this the left mechanisms to prevent the dsRNA formation. So as just mentioned, we have built up these platforms. So for this T7 RNA polymerase, we applied 3 of them. First is FADS and MTPS for small library screening, and also the computer-aided residue design to have hotspot identified using this calculation, so that we can do less screening. And so here is how we design the fluorescence using the FADS in order to use our ultra-high throughput screening platform, FADS. So as just mentioned, we want mainly the full-length RNA instead of abortive. So if we can correlate the fluorescence identity with the full-length RNA content, we will spot out the variant with more full-length RNA content. So we designed a molecular beacon at the end, the 3' of the template. And also we also see that if there are loopback and the fluorescence intensity is decreased. So we have hypothesis that if we put the molecular beacon at the end of this template, we will spot out from the FADS screening the variants with higher activity to produce more full-length RNA. So here's how we do the FADS. Basically, FADS is to put each single mutant to one cell, so to one droplet. So we put the lysozyme and the cell together, and then we put at most one single mutant, which is also a single cell, in a droplet in picoletter. So after incubation, which we do the IVT offline, and then we do the sorting. The one with the higher fluorescence intensity will be separated with others based on our fluorescence threshold setup. So we firstly sequenced the dominant mutate collected from the positive channel. So the positive droplet DNA was amplified and then sequenced using NGS sequencing. So we can see some of the mutations. Some of the mutations are enriched in our system. And later we identified individual mutants using the separation and also the culture outside. Yeah. Then we identified some mutants with decreased RNA. And we also saw some mutants with decreased yield. Maybe this is not the screening pressure, so it's not a surprise. We will fix it later. So we also applied rational design, as just mentioned, from the mechanism of the dsRNA formation. So if we have less abortive RNA, we will have less dsRNA as well. So we have a hypothesis that if we can lower the folding free energy of the elongation chain phase, elongation from a conformation, maybe the conformational change will happen easier and also will be stable. So we did the calculation of this free energy changes and spot out 10 mutations for saturation mutagenesis listed here. And we also found out that the ones from the calculation, the mutations from the calculation, the top 10, also most of them, none of them located at the conformational change areas. We can see from here, these balls. So in order to do this screening for this saturation mutagenesis, and also to avoid yield sacrifice, so we designed another screening platform, macrotide screening, using 2 probes. One is the same to the FADS, which is at the end of this template. And the other one, the new one, we put in the beginning of the template, the 5' probe, CY5 probe, so that it will help to monitor the yield, the total yield of the RNA synthesis. So using this macrotide screening platform, after several rounds screening, and we found 2 mutants with decreased dsRNA content and ensured yield. The third step, so because we identified several beneficial variants, so we did a round of DNA shuffling from these previous libraries, the good ones. We combine them and find a new mutant, mutant 17. It has less dsRNA than all the 3 parents, and also it's only about 2% of the wild type. So it's really significantly decreased. So we also want to know why and how come. So it's really impressive. So based on our hypothesis, we tested 2 different site activities. One is the terminal transferase activity, and the other one's RNA-dependent RNA polymerase activities. And the results showed that it correlates very well with the dsRNA content. That means this dsRNA content decreasing is attributed mainly to these 2 site activities decrease. We also did some other tests, for example, the yield, the mRNA integrity, and also the capping efficiency. And the data showed that none of the 3 of other properties were not sacrificed either, with significant decreased dsRNA content. The immunogenicity also tested, and so it also correlates very well with the decreased dsRNA content. This data shows that the expression level is not sacrificed or decreased using the EGFP protein expression system. So this result is really impressive. So we also drafted a paper. It is now preprinted online. You can search, and if you want to learn more about how we involve this enzyme, you can read online. And we also filed a Chinese and US patent. Most importantly, we have launched a new T7 RNA polymerase based on our best variant. It's called Clear Script. And when we compare the new product with our old product, which is the wet type, and also the best dsRNA, low dsRNA product on the market, and we can see that our new version T7 RNA polymerase has extremely low dsRNA content, with only 22% of the wet type and 19% of reference. As I just mentioned in the beginning, so if we don't have low dsRNA T7 polymerase, we need to do a lot of purification. So this data shows that even using our new version T7 polymerase, you don't need to do the cellulose treatment. And the dsRNA content is also lower than that when you do this purification. This shows the beneficial of this new T7 polymerase. So here is our product. This is a research grade, and our GMP grade mutant will be available soon, maybe next month. Okay, here the update. Thank you, and feel free to ask any questions online or offline. Thank you.
Thank you.
Hi, Kefan. Yeah.
Hello. Yeah. Yeah. Hello. Yeah. Hello.
Yes.
Yeah. Yeah. Thank you. Thank you, Dr. Liu, for your insightful presentation. And we now open the door for questions. Please submit your questions through the Q&A tools. And now there is already some audience has left the questions. And the first, Dr. Liu, can you also see the Q&A questions?
Oh yes, I can see.
Yeah. Okay. And the first question is, does the T7 mutant yield have any relationship with the reduction of dsRNA? How is it achieved that the yield is not reduced? Thank you.
Oh, the first one. So, we do see some trade-off at some point for the. So if we get most dsRNA decreased variant has a lower yield, but we also found some of them with both properties improved. So I think the relationship is not really a trade-off, but still we are still working on the higher integrity in T7 RNA polymerase, and we still haven't got it yet. Maybe the three property together, maybe there would be some trade-off, some relationship. We will see. We are now working on it. Maybe later on we will publish a new variant with higher integrity. Okay. So how is it achieved? I think that's evolution made it. It's nature, yeah.
Okay, thank you, Dr. Liu. And the other questions you can also see. Yeah, you choose one.
There are many questions.
Yeah, you can choose one to answer.
Yeah, so maybe the molecular beacon work is patent. Not really. We use the published ones. Yeah. What is the current level of reduction in double-strand RNA? I think this question Kefan can answer.
Which one?
The current level of the double-strand RNA. I think it's in my presentation. There's the data, but when we use different template, the data change a little bit, I would say. So here is, I think the level is still here. It's 0.007, yeah, nanogram per microgram. If this is a question and this is an answer. So back to the question. How is the time, Kafan?
There is 8 minutes left. Yeah, still 8 minutes. Yeah.
Can the link to the literature be sent in the chat box? I can do it very quick now. The literature link, I think is here. Okay, here the answer. So another question is about how the quantity of enzyme in HTS, you mean HTPS, should the protein amount be normalized? Are all other steps this? So here the question, Liu Yi. No, not really. For the MTPS, for the screening, we don't measure the amount. We ignore the A-pricing level differentiation. That is high throughput screening. Additional, should RNA and DNA be removed before applying the beacon? We don't remove this because this the beacon assay we did in vitro or in vivo. In vivo, we didn't do any removal of RNAs and DNAs. And in vitro, we did some purification of these enzymes, but the RNA and DNA from elsewhere, we didn't isolate, we didn't remove, because it's really specific for our template. So I think it's not a problem. How can the pipelines of directed evolution and residual design be combined? How many mutations have you screened? Wow, it's a good question. You can find the answer from the paper. We wrote down the whole process, even the failure part. How we combine? Actually, I talk about the two screening platform. One is the FADS, one is the macrotide plate screening. In real screening, we do the FADS, and then the identified ones will go to macrotide plate screening. So it's like firstly macrofluidic screening and then macrotiter. But for small small libraries such as the saturation mutagenesis, we only do the macrotide plate screening. Because it is dual probe. It has two probes and will give a signal about the yield, so it's more accurate. The pressure is it has two pressures, screening pressures. How many mutations? I didn't remember, because for the FADS, each round it's above 1 million. So we did at least, I didn't remember, several rounds. So it's about million scale. But because our we use the error-prone PCR, so the actual mutations or variants maybe not that high. And also because for each round, we can screen 1 million droplet, but it has only 10%. Only 10% of the droplets has one single mutant, because 90% is empty. To make sure, yeah, each droplet only has one single mutant. That's mathematics, the frequency. Other questions? What dsRNA standard used for dsRNA quantification? Maybe I can answer later, because we used what standard? Maybe 2K or 4K different kind of template, actually. Maybe later this question we will dig out our data. Can your customer use ClearScript for clinical use without paying patent fee? I think the answer is yes, because we sell products, and you should have if you buy the product, you can use it free, if I'm right. There's another friend talk about the details about the GMP standard. So can you please leave your contact information, and our standard can be sent through email. We have already produced more than 10 mRNA related enzymes in the past, including the wet type T7 RNA polymerase. This is the upgraded new version. It follows the same standards as previous version, and as well as other enzymes. Don't worry.
Okay. Okay, thank you.